Hey everybody, welcome back to another RevWise video. Today we have for you something very special. We have an A-level paper. This is our first time doing one on the channel. Decided to switch it up, right? Because I know a lot of A-level people in my uni and I want to see how difficult these A-level papers are. So we're going to do the full walkthrough, okay? Or just solve the paper along with me. I'm going to explain all my answers, explain how we got to them. And yeah, we'll see how challenging the paper is compared to IB. So let's go right into it. So, question 1. Figure 1 shows the structure of a fatty acid molecule and the structure of a glycerol molecule. So we have our fatty acid and we have our glycerol, okay? And then it wants us to draw a circle around the part of the fatty acid molecule and part of glycerol that is removed to form a bond in the triglyceride molecule. And it wants us to name the type of bond that is formed and name the reaction. Okay, easy question. So, off the bat, they want you to circle bonds. They're not going to make it that counterintuitive for you. They're going to circle the regions that form the bonds are going to be close to each other geographically on the paper, right? So it's something from here, something from here. You can already narrow that down. Now, in terms of bonds that you know, we often have to take an H and an OH, right? And then if you don't know this reaction, which you really should know if you're doing your A-levels, but if you don't know this reaction, it's kind of process of elimination, which H, which OH you take, but it should be pretty clear, right? So we have an OH here, so it makes sense to take the OH from our fatty acid molecule, right? And now for the H, we don't want to take, one OH from this fatty acid can't react with all three of these exposed ones in our glycerol, so we're going to pick one of them. I'll pick the middle one, and we're not going to circle the OH, we're going to circle the H only, because that's what's going to take part in this reaction, this specific type of reaction, which we will name very shortly in the next question. The type of bond here is a special type of bond called an ester bond, right? An ester bond, I'm sure you're familiar with that, because it's what I'm taught, that ester bond is formed in between the fatty acid and glycerol in the triglyceride. And what type of reaction is it? Well, if you're being quite literal, it's an esterification reaction, but I'm guessing that's not what it wants us to say here. The general type of reaction that's occurring because of the manner in which water is being formed, we get a condensation reaction, right? Condensation reaction, right? Because we have this OH and H, and then, yeah. So this is a condensation reaction. Lovely. Okay, question number 2. Name the fatty acid shown in table 1 that is a saturated fatty acid, okay? So saturated fatty acid, we're going to look for all single bonds. If we see a double bond, we know it's unsaturated, right? It could be polyunsaturated, monounsaturated, but we know it's unsaturated. So it's pretty clear which one only has single bonds. It's this top one, stearic acid. Okay, the melting point is the temperature at which a solid changes to a liquid. Use table 1 to describe the relationship between fatty acid structure and fatty acid melting point. Okay, this is not even, this is just using the data based on what you see, right? So we can see that we have a polyunsaturated fatty acid in the linoleic acid, right? We have multiple double bonds, and then we have a monounsaturated fatty acid with oleic acid, has one double bond, and then stearic acid, as we described, has no double bonds. And we're seeing melting point is going from high to lower to even lower. So based on this, it's quite clear that as we increase the number of double bonds, the melting point is decreasing. So how do we write this? So I'm just going to write it in very simply. Don't write like this in your exam. I'm just writing it because I don't want to spend too much time writing during this paper. So as we increase the double bond, right, then we see a decrease in melting point. And it's one mark that will be good enough. Okay, the ratio of saturated to unsaturated fatty acids in a cell surface membrane determines the extent to the membrane's fluidity. Now, scientists provided a cell culture of mouse phagocytes with a liquid broth rich in unsaturated fatty acids, and they observed that an increase in the proportion of phospholipids in the phagocytes containing increase in the proportion of phospholipids in the phagocytes containing unsaturated fatty acids, and they also observed more phagocytosis. And they want us to explain why this would happen, right? So they put these mouse phagocytes in a culture full of unsaturated fatty acids, right? So these phagocytes, they have a membrane like any other cell. And what's going to happen if you put them in a liquid broth rich of these unsaturated fatty acid chains, it's going to incorporate them into its membrane, right? And then that explains why we're seeing an increase in the proportion of phospholipids in the phagocytes containing unsaturated fatty acids, right? So, why there was more phagocytosis? Well, what is the job of a phagocyte? So we know the phagocyte is basically engulfing cells, right? What would be useful if you're engulfing cells? It would be useful if you're able to wrap around the cell more easily, right? If you're engulfing cells, it would be great that if the membrane was very flexible and able to move, right? So we know that we incorporated more unsaturated fatty acids. And we know the membrane is likely more flexible, which is why we're seeing more phagocytosis. So what's the logical conclusion? The logical conclusion is that the incorporation of unsaturated fatty acids made the membrane more fluid. It increased the fluidity, and this increase in fluidity allowed greater phagocytosis due to the membrane being more flexible. So how would we word this? Well, an easy way to think about it is just to think of the 3 marks, right? Make sure you have 3 points in here, right? So we're increasing the unsaturated fatty fatty acids, right? And that's increasing membrane fluidity, right? Now this increase in membrane fluidity makes the phagocyte membrane more flexible, right? So we've established the 2 important things. Now, what's the conclusion of this increased flexibility? It makes it better, more efficient, more adept at engulfing cells, right? And this leads to higher phagocytosis, what we observed. Very nice question, easily done. Describe the processes of facilitated diffusion and active transport. 3 marks, you should know this pretty easily. Okay, facilitated diffusion, remember, facilitated diffusion is quite similar to passive in terms of it going from a high to low concentration, right? You're not moving against the concentration gradient, right? So it's down a concentration gradient. But the key difference between facilitated and passive is the idea that facilitated is via a carrier protein. It's actually a carrier or channel protein. Carrier or channel protein. That's all it is. That's all it is. It's quite a simple thing. Now, active transport is even easier to say because we know it's against a concentration gradient. So up a concentration gradient from low to high. And this is always with a carrier protein, right? A channel protein can work for active transport because you need ATP to be able to move it against the concentration gradient. A channel is just simply facilitates this passive diffusion across the channel. So you need a carrier protein and you need to use adenosine triphosphate, ATP. Okay, lovely. What are microvilli? Very, very broad question, right? How would you even word this? I think I would just say invaginated finger-like. I won't even say finger-like. That's more for regular villi. Invaginated projections on a villi surface, right? How else could you say it? They're quite literally folded, highly folded parts of a plasma membrane, right? That's what you're actually seeing. These those parts of a microvilli, that's just folds basically. So, I think folded parts or invaginations in a plasma membrane, however you say it, quite an easy way to. Okay, vitamin A is a fat-soluble substance. Micelles are formed involved in the process of vitamin A absorption. Describe the process of vitamin A absorption into the cells lining the ileum. Okay, there's quite a structured response. First things first, whenever you see fat-soluble vitamin and it wants to talk about absorption, the key is bile salts, right? What bile salts do is they essentially mix with vitamin A, and what the effect of this is vitamin A becomes more water soluble. Now, the theory behind how it becomes water soluble is basically the bile salts are emulsifying the fat. This forms micelles, right? Which I'm sure you know, they're like tiny droplets, and they're essentially keeping the vitamin A dissolved in the intestinal contents, okay? So at this point, the micelles are essentially the delivery system of the vitamin A. So they're bringing the vitamin A to the intestinal cell surface. So micelles bring vitamin A to intestinal cell surface, right? Like basically the enterocyte membranes, right? They go really close to these enterocyte membranes, which are in the ileum and also earlier regions of the small intestine, and they keep a high concentration of vitamin A near the cell surface. And this is important for diffusion, right? As you're keeping this high concentration of vitamin A near the membrane, what's going to happen? Well, diffusion. So vitamin A is going to diffuse into cells, right? Just through the cell membrane, right? Passive diffusion. And then further absorption occurs further down, as we know. So yeah, pretty nice question. And we're on to number 3. Describe how we breathe in. You know, A-level questions are quite funny. You know, IB is quite specific, and then A-level you just got statements like this. Describe how we breathe in. I mean, fair enough, you know. But okay, so just the process of inhalation. So some key things to mention here. The idea is you want to mention something to do with your ribs, you want to mention something to do with your diaphragm, and you want to do something to do with thoracic pressure, right? Those are the key points you want to go with. Those are the three main things that are involved in inhalation, okay? So let's start with the ribs, right? So in our ribs, we know we have these intercostal muscles, and we have these important ones called external intercostal muscles. What do they do? Well, these move our ribs upwards and outwards, right? So they contract and our ribs go. So they contract and then our ribs go up and out. Okay? First idea. Second idea is our diaphragm, right? Which we know is this part at the bottom of our lungs, right? Our diaphragm, it flattens. So it's contracting and it pulls it down. So diaphragm contracts, pulling in. So we've made our ribs go out and we made our diaphragm go down. What does that mean? Well, we've increased thoracic volume, right? Now we've increased thoracic volume, but the amount of particles in our thorax are the same. So what does that mean? You increase volume without changing anything else. We have decreased thoracic pressure, right? And this is what allows air to be forced into our lungs. Lovely. Scientists prepared alveolar tissue to view using an optical microscope. The scientists cut very thin slices of the alveolar tissue. Explain why the scientists used very thin slices of alveolar tissue with the optical microscope. Good question, right? So why would we only want a very thin layer? Well, the idea is when you're using a optical microscope is you want the light from the microscope to be able to go through the cell. So if you have a very thick layer of tissue, what's going to happen is the light won't be able to penetrate the tissue. You won't get a good view of what you're looking at, right? So explain why. So the scientist, he took, let's say, a few layers of alveolar tissue so that light can pass through the cells. right? It also makes it easier just to visualize what you're looking at. It's less clutter, but this is the main idea, right? There we go. Figure 2 is an image of the lung tissue observed using an optical microscope. So there we go. And it's labeled A and alveolar epithelium, and it wants us to identify the tube labeled A, right? Okay, so we can see A, it has a very distinct branching pattern, which I'm sure we know of, and we know that if that's the alveolar epithelium, what's the tube-like branch that's coming into the alveoli? We should know it's a bronchiole. Bronchiole, remember, are those dichotomous. They're those branches that come off the bronchi and increase the surface area of the lungs by a lot, along with the alveoli. Scientists use a ruler to measure the diameter of some of the alveoli. Table 2 showed the scientists' results. We have a table of the alveolar diameters, and then it wants us to, so we know the magnification is 40x, and it wants us to calculate the mean diameter in micrometers of the alveoli. Okay, so the formula we're doing is the real size is going to equal the size we see in the image, which is the table values, over the magnification, which is 40. So it's something over 40, and then we just got to calculate the mean of these. So it's 4 plus 2 plus 5 plus 1 plus 2 plus 3 plus 5 plus 2, which is 24. And then there's 8 numbers, which is, 3. So it's 3 over 40, but that's 3 millimeters, right? Let's just do this. So 3 over 40, 3 tenths. So it's 0.075, and that's millimeters, right? So then we make it micrometers, we get 75 micrometers. There we go. Give the uncertainty associated with taking a measurement using a ruler with 1 millimeter graduations. Calculate the percentage error for a measurement using the ruler of 4 millimeters. Okay, so 1 millimeter graduations, that means that our raw uncertainty is going to be 1 millimeter, right? And then our percentage error, if we have a ruler measurement using the ruler of 4 millimeters, and we know our uncertainty is 1, it's 1 over 4, 25%. I guess you could say 0. Oh, one's percentage, right? So 25%, yeah. Okay, nice. Molasses is a solution obtained from sugar beet plants. The sugars present in molasses are sucrose, glucose, and fructose. Give the number of different types of monosaccharides present in molasses. Wow, very interesting. Okay, molasses is what's in brown sugar, right? So, yeah, brown sugar is just white sugar with molasses, if that helps you remember what molasses is. But essentially it wants to identify that glucose and fructose are our monosaccharides, and then sucrose is our disaccharide. Yeah, so the number of different types of monosaccharides are 2. A student used a biochemical test for reducing sugars on a clear sample of molasses. Describe the biochemical test for a reducing sugar. Explain the result expected from the test on the sample of molasses. Okay, so it wants the biochemical test. Oh, for reducing sugar. Okay, so it wants us to talk about Benedict's solution, and then the result. Okay, so the description of the test. So we have to heat our sample. Sorry, I just wanted to see what I wanted to call it. Heat sample with Benedict's solution. Right, and what happens? Well, we know that due to the presence of our sugars there, we're going to see red precipitate formed, right? And the reason we have our monosaccharides, you can mention either of them, but our reducing sugars, glucose or fructose, glucose is a reducing sugar. Okay, explanation of the result. Well, because our reducing sugars, glucose / fructose, are present in the sample, I guess. Yeah, that's the explanation. I mean, don't know what I want you to say there. Okay. Free sugar is the sugar in food and drinks released when food is crushed or when sugar is added to food at home by manufacturers. Scientists recommend that no more of 5% of energy consumed per day should come from free sugar. The main daily energy requirement for a 10-year-old is 8,100 kilojoules. The free sugar in 1 tablespoon of molasses contains 250 kilojoules of energy. Calculate the number of tablespoons of molasses required for a 10-year-old to reach the recommendation for energy consumed in a free sugar per day. Wow, that is a strangely long question, but quite easy. So we know that no more than 5% should come from free sugar. So then we know the child's daily energy expenditure is 8,100. So 5% of that, we have 405 kilojoules, right? So we're looking to get 405 kilojoules as the upper limit. And we know that the free sugar in 1 contains 250. So 250 times 2 is 500 kilojoules. It's already over. But I guess the number of tablespoons, we're gonna have a decimal number, right? So how do we do that? We're just gonna divide it by 250, right? So that's the number that of free sugar the child's allowed to have per day. This is the energy in a single tablespoon of molasses. And let me just plop this in the calculator real quick. So 405 divided by 250, we get 1.62 tablespoons. So you're only allowed to have 1.62 tablespoons of molasses. So be mindful. A scientist used the apparatus in figure 3 to investigate osmosis. Okay, so let's see what we have in figure 3. We have air in the tube here. We have a lot of molasses in this tube. We have some water, and we have an animal's bladder. And it wants us to use our understanding of osmosis to explain why the air pressure in the tube increased. So we have a certain amount of air molecules here, air pressure increased. That means that the volume that the air occupied decreased, right? So the real question is, why would the volume that it occupied decrease? So let's think about this. What could it be? Okay, yeah, makes sense. So the question's about osmosis, right? And then we have the animal's bladder, which is acting as our partially permeable membrane. And then we have 2 things with different water potential. I think you can see where this is going. Okay, first idea that you want to note is that molasses has a lower water potential than water, right? Who would have thought, right? Than water. Okay, so when you have something here that has a higher water potential and you have molasses with a low water potential, what's going to happen? Well, water's going to want to try and come inside, right? By the laws of osmosis, it's going to pass through this partially permeable membrane and it's going to go to the lower water potential. So water will pass through the bladder, right? And this is going to be into the tube. Now, you're increasing the volume of a liquid in this tube by the water joining the molasses, and that's going to leave less and less air, less and less room for the air, right? Because in terms of quantity, the air is the same, but you have less volume, that's going to be increased pressure. So, how do we say this? Well, decreased air volume, right? Therefore, increased air pressure. There we go. Lovely. The scientists repeated the investigation but made one change to the molasses. The scientists did not change the volume of molasses at the start of the investigation. The scientists observed that the air pressure in the tube increased by 160 kilopascals compared with 800 kilopascals in the first investigation. Suggest the change the scientists made to the molasses to create the smaller increase in air pressure. Use the air pressure figures in a calculation to support your answer. Okay. Now, what's a logical thing we can do? We established that the reason this occurred is because of the disparity in the water potential between molasses and water. So how could the scientist change the disparity? Well, he could have just diluted the molasses. He could have added water to the molasses. So how do we suggest it? Do we just suggest it here? Oh yeah, here. Okay. So he decreased the concentration of the molasses said molasses too many times per day, this question, I swear. And it wants us to use a figure in our use a number. I guess it just wants us to use like a percentage or ratio. So I mean, it's just like 160 times 5 is 800, right? So this one is 5 times more than, yeah. So how do we say that? If the ratio increased like this, right? So let's establish that 800 over 160 equals to 5, right? Yeah, that's right. So 5 times reduction, and to support the answer, I wonder if it wants us to like suggest that the 5 times reduction in air pressure is going to directly correlate with him diluting the molasses by a factor of 5 times, because I don't think that would be accurate, right? No way that if it would correspond exactly like that. I think this is fine. I think if you just say there's a 5 times reduction in air pressure, right? That's fine. If you want to make claims on the dilution of the molasses, I think that could work too. Say there's he increased the concentration. He reduced the concentration of molasses by 5 times. That works as well. However you want to do it, but as long as you're getting this number of 5 from there, that's a calculation. That's the air pressure. Yeah. Just say there's a 5% reduction in air pressure. That should do you fine. Okay. Describe the appearance and behavior. Oh, finally we're done with molasses. Can I just say that was too long. And behavior of chromosomes during prophase and during anaphase of mitosis. Oh, lovely memorization question. Very easy. Let's do prophase first. So in prophase, what do we? Oh, appearance and behavior. Okay, so we have these sister chromatids. We have these or chromosomes, and in prophase they're condensing, right? In terms of appearance, they're visible. Yeah, pretty fair things to say. Anaphase, remember anaphase is where they're splitting apart, pretty distinct aspect. So what can we say? Well, the centromere, the chromatids are pulled to opposite poles, right? If you didn't know, we have 2 poles in the cell. We have a animal pole and a vegetal pole. So opposite poles, the animal and the vegetal pole. And the reason I wrote centromeres earlier, I was thinking of the centromeres are divided, right? The centromeres become split, and the chromatids are pulled to opposite sides, and yeah. Okay. 5.2, it seems that they used a figure that's not here, and we have a website. Yeah, I'm not going to that. We're just going to skip this one. I can't lie. I don't think that would be too hard. What is it one? Mitotic index? Yeah, mitotic index, you're just counting, right? You're counting. I'll give you the formula if you... So it's total cells, and then over it is cells undergoing mitosis, right? So cells undergoing mitosis over total cells. Okay, 5.3. The scientists used data from figure 4 to create a R value. The scientists then used statistical tests to determine the probability associated with the value of R. So R equals minus 0.98, and then P is less than 0.05. Okay, so we have the R and P values. So what can you conclude from the result? We can say that there is a negative correlation. Remember, the R value shows correlation, and then the closer it is to 1 or minus 1 shows the strength of the correlation. In this case, it's very close to minus 1, showing it's a strong negative correlation, right? And then we know that this is significant because P is less than our alpha. Remember, our alpha is our significance level, the value we set. So we just chose this level of 0.05. We could have put P is less than 0.01 if we wanted to be more thorough, or yeah, so on and so forth. You could make it whatever you want. But generally in science, we use P is less than 0.05. And then this means that it is significant, and that allows us to reject the null hypothesis. Okay, what can you conclude about the effects that what can you conclude about the effect that distance from the root tip has on the proportion of cells in different stages of the cell cycle? Use information in figure 4. Well, we can't see figure 4. Can we work this out without figure 4? It wants us to use information from figure 4, so I don't know. Yeah, I'm trying to think of how we would do this. Yeah, we can't really answer that. We kind of need to look at the thing if we want to